students. Today we are going to discuss the fourth lesson of your biology, that is excretion, the waste disposing system. So, you know what we learned about different metabolic activities from lesson 1, lesson 2. So, what are the different metabolic activities like photosynthesis, digestion, respiration? These all are metabolic activities takes place in all living beings. So, by the result of these metabolic activities, some useful products are released, means like glucose, even energy is released. At the same time, along with the useful products and energy, even for every metabolic activity, there is the release of some waste molecules, or waste materials, or unwanted materials. So, here the excretion is the biological process which is associated to separate or to shift the waste molecules from the body. So, excretion is the biological process for separation and removal of waste molecules from the body. So, here the term excretion came from the Latin word. In the Latin, ex means out, genera means shift. Means shifting the waste molecules out of the body. That we call as excretion. So, here what are the different waste molecules released in the human? For example, we release waste molecules like CO2, water in the process of respiration. Even most of the metabolic activities in the human body release nitrogenous compounds as their wastes. For example, nitrogenous compounds example urea, uric acid, ammonia, bipigments, even some excess salts. So, whenever the salts in your body are maintained in the normal level in the body fluids, then only the proper functioning of vital organs takes place. But when there is excess salts, it leads to the imbalance in the homeostasis. It leads to the disturbance in the homeostasis of the body fluids. That again shows the impact on the functioning of different organs. So, we always have to maintain the proper balance of salts and water in the body. So, here the function of excretory system in the body, the main function of the excretory system, simply not only remove the waste molecules from the body, at the same time maintain the homeostasis, a balance of different body fluids in the body also done by excretory system. So, coming to the living organisms, especially animals, different organisms, different animals develop different types of excretory systems. So, in highly developed organisms, there is a specific system like excretory system. So, now we go with the human excretory system. Students, all of you just watch this diagram. What are the different parts of the human excretory system? So, just look at this diagram. If you just observe the diagram carefully, understand about how the excretory system of human. So, coming to human excretory system, there is a pair of kidneys and a pair of long narrow tubes known as a pair of ureters and urinary bladder, urethra. So, our excretory system mainly contains 4 parts. There is a pair of kidneys, means right kidney and left kidney, a pair of ureters and urinary bladder, pear-shaped satellite structure, and a narrow outlet known as urethra. These are the 4 main parts of our human excretory system. So, coming to human excretory system, I already told you what are the different parts. So, now we will see what is the main part of the excretory system is nothing but human kidneys. So, when we talk about kidneys, the kidneys are a pair of bean-shaped. Have you seen this is bean-shaped structures, and these are reddish brown in color, bean-shaped reddish brownish structures present in your abdominal cavity on the either side of, and they are arranged on the either side of your vertebral columnar backbone. So, if you observe the kidney's internal structure again, The outer portion of the kidney is dark green color. We call it as cortex. And the inner part of the kidney we call as medulla. So the outer region of the kidney we call as cortex. The inner region of the kidney we call as medulla. And here inside the kidney again there is a inverted pyramid like structures are present. We call them as renal pyramids. What we call means we call them as renal pyramids. And these renal pyramids again they opens into the structures we call as calces. What we call calces. These calces and renal pyramids opens into a Y-shaped opening of the kidney we call as pelvis. Calces and renal pyramids together they lead to the pelvis. The pelvis through the fissure or notch-like structure of the kidney. This one what we call means hilus. What we call hilus. Through the hilus the pelvis opens into the tube-like structure known as ureter. Okay now? And so here there is a two special blood vessels known as renal vein and renal artery are present. So here through the hilus of kidney, renal artery enters into the kidney, and at the same time through the hilus, ureter and renal vein will be exit. Means renal artery enter into the kidney through the hilus. Means this notch-like structure. Means this one we call as actually the opening of kidney we call as hilus. So the renal artery enters into the kidney through the hilus, and this one actually what we call this one means the renal vein and even ureter also exit are coming out of the kidney through the hilus only. Okay now. So here the renal artery brings the oxygenated blood to the kidneys, which is having waste molecules or waste materials present in the blood. The renal artery brings oxygenated blood with the waste molecules in the blood. Once the blood is brought by the renal artery, with the waste molecules in the kidneys, actually the blood is filtered in the kidneys. The waste molecules are separated or filtered out of the blood, and the purified blood or the filtered blood again through the renal vein it will be coming. Okay na? So here the renal artery, sometimes in the bit they ask which blood, either through renal artery or renal vein. The oxygenated blood with the waste molecules enters the kidney means through the renal artery, and the filtered and purified blood from the kidney is brought by which blood vessel means through the renal vein. So once the kidneys are over, and coming to measurements of kidney, if you observe each kidney in the lengthwise it is nearly 10 centimeters in length and 5 centimeters in breadth, and it is 4 centimeters in thickness. And each kidney consists of a million, nearly 1.3 to 1.8 million number of small tiny tubules like structure. What we call the tiny tubules means nephrons. So even the nephrons are also called as uriniferous tubules. What we call uriniferous tubules. And the uriniferous tubules or nephrons we called as the structural and functional units of kidney actually. Why? Because the urine formation, which is the actual function, takes place in the kidneys, occurs in the nephron of the kidney, and each kidney again contains a million number of nephrons. That's why the the nephrons we call as the structural and the functional unit of kidney. Most of the time they ask in the bit what are the structural unit of excretory system or what are the structural and functional units of kidneys means the answer is nephron. Don't get confused. Nephron is the structural and functional unit of kidney and neuron is the structural and functional unit of brain. So once the urine was formed in the nephrons, that nephrons, the million number of presents in each kidney, these all nephrons together form renal pyramids and calyces. So the urine was enters into the pelvis of the kidneys. From that it will be entered into the long tubules known as ureters. So these ureters are the narrow tubes, white color, narrow distensible tubes, means they are able to stretch themselves, means they are able to adjust their volume according to the volume of the urine. So the ureters are the long, thin, narrow white color distensible tubes which are originates from the each of the kidneys, and the length of the ureter is nearly 30 centimeters in their length, and the urine from the kidney will be enter into the next structure known as urinary bladder through the ureters, and in the ureters the urine was passes by a movement known as peristalsis movement, which we already discussed in the first lesson nutrition, how the food was passes in the esophagus or food pipe by the wave-like movements known as peristaltic movements. In the same way, in the ureters also the urine passes by the peristaltic movements, and from the ureters the urine will be entered into the urinary bladder. The urinary bladder is a pear-shaped sac-like, it is also distensible sac-like structure, and the urine from the ureters enters into the urinary bladder. In the urinary bladder it is temporarily stored, and the total capacity, the storage capacity of the urinary bladder is nearly 300 to 800 ml of urine will be stored by the urinary bladder. And from after the urinary bladder, a tube-like structure, narrow opening through which urine will be excreted by, we called it as urethra. So where the urinary bladder enters into the urethra, it was surrounded by two sets of muscles or sphincters. So these two sets of muscles, muscle rings or sphincters help to control the excretion or the elimination of urine to the out. And urethra, it is the part through where urine is passes, and urethra, it is in females it is 4 centimeters in length, and coming to males it is 20 centimeters in length, and this is the external opening. And have you seen like small cap-like structures are present over the two kidneys? Actually these cap-like structures are known as adrenal glands. These two cap-like structures are known as adrenal glands. These adrenal glands also release different hormones which plays major role in our metabolic activities, and at the same time okay, and before going to explain about nephron, what are the next structures, okay, first we go with the structure of nephron. So listen carefully. Here I drawn a small rough diagram of the nephron. I already told you kidneys contain a million number of nephrons. So just imagine this is the one single nephron structure. So if you observe the nephron structure, it mainly contains two parts. One we call as Malpighian body, and the second one we call as renal tubule. First one we call as Malpighian body, and the second one we call as renal tubule. So again the Malpighian body of the nephron contains two parts. So here the opening of the nephron, which is cup-shaped like structure, this cup-shaped like structure of Malpighian body we call as Bowman's capsule. The broad end of the nephron, which is like a cup, that cup-shaped broad end of the nephron we call as Bowman's capsule. In the Bowman's capsule, there is a network of blood vessels. That network or a bunch of blood vessels we call as glomerulus. So glomerulus plus Bowman's capsule together made the Malpighian body. So the broad end of the nephron we call as Bowman's capsule, and the bunch of network of capillaries we call as glomerulus. Glomerulus and Bowman's capsule together made the Malpighian body. And the remaining part of the tube-like nephron part, what we call is renal tubule. Again, the renal tubule contains three parts. The renal tubule contains how many parts? Three parts. First one we call as PCT. Second one we call as loop of Hilli. Third one we call as DCT. Okay. Okay, now the renal tubule again contain PCT, loop of Henle and DCT. PCT means proximal convoluted tubule, and the U-shaped portion of the nephron we call as loop of Henle, and the after loop of Henle again the part of the renal tubule we call as DCT and distal convoluted tubule. This DCT finally opens into the collecting duct. Actually, in the nephron, these all collecting ducts together form renal tubules and cases. That renal tubule and cases opens into pelvis and finally opens into the ureters. Okay, na? And here the structure of nephron is very important. Why? Because actually the urine formation takes place in the nephrons only. Na? So now we try to understand actually what happening in nephron. Okay, na? So in the formation of urine from the blood, it mainly includes 4 stages. The first stage of the urine formation we call as glomerular filtration, and the second stage we call as tubular reabsorption, and third one we call as tubular secretion, and the fourth one we call as concentration of hypertonic urine. So what actually happened in every stage we see that. First one, glomerular filtration. Actually, before going to explain about glomerular filtration, here the renal artery divides and it will be entered into the Bowman's capsule in the form of afferent arteriole. The branch of the renal artery we call as afferent arteriole. This afferent arteriole enter into the Bowman's capsule and divide into many capillaries, form glomerulus, and will be coming out in the form of efferent arteriole. These two are very important. The portion afferent means goes in, means it enters into the glomerulus. Such arteriole we call as afferent arteriole. And efferent arteriole means which comes out, means from the glomerulus, the portion which comes out, in which form it comes out we call as efferent arteriole. So here come back to the afferent arteriole. The diameter of the efferent arteriole is less, means the diameter of the efferent arteriole is less, means it is very narrow. So because of the narrowness of the efferent arteriole, some pressure will be exerted in the glomerulus. At the same time, the wall of the Bowman's capsule is lined by a single layer of cells we call as podocytes. What we call podocytes. The Bowman's capsule inner lining is made up of single layer of epithelial cells. We call the cells as podocyte cells. These podocyte cells leaving small gaps or small pores between them. Because of the small pores, the blood is filtered out of the glomerulus and will be entered into the Bowman's capsule. Means because of the narrow outlet of the efferent arteriole, some pressure will be produced in the glomerulus and bunch of blood vessels. So because of the pressure, some pressure filtration, and even we also call that filtration as ultrafiltration. Because of the ultrafiltration, some molecules from the blood will be filtered out, means filtered out from the glomerulus and enter into the Bowman's capsule. And what the filtrate from the glomerulus enter into Bowman's capsule we call as glomerular filtrate, and even it is also known as primary urine. So the primary urine is, the composition of primary urine is almost equal to the blood plasma but except some blood cells. Okay, na? So it means most of the molecules will be filtered out and present in the primary urine. But if the primary urine passes in the same way, is it not benefit to our body? So even in the primary urine, many useful mineral salts, vitamins, even most of the amount of water is still present. So the second step we call as tubular reabsorption. So the tubular reabsorption takes place in the PCT of the renal tubule. So in the tubular reabsorption, what are the useful molecules like water molecules, water molecules, even waste molecules, these all will be entered from the renal tubule into the peritubular network. Means the efferent arteriole will be again divided into many branches and surround over the renal tubule part of the nephron. That this one we call as pericapillary network or peritubular network. This pericapillary network, peri means around, means around the nephron, the part of the capillaries which are present will be called as peritubular network. So in the PCT region, the tubular reabsorption takes place. Means what is the excess water and even some useful molecules should be reabsorbed back into the blood capillaries or reabsorbed back into the peritubular network from the PCT region of the renal tubule. Even nearly 75% of water is also reabsorbed back in the PCT region into the blood capillaries. And the next one we call as tubular secretion. So once the primary urine reabsorption takes place, it will goes along in the loop of Henle and finally it enters into the DCT region of the nephron. In the DCT region, in the distal convoluted tubule, so the waste molecules from the blood capillaries will be secreted into the DCT region of the renal tubule. So just observe the difference carefully. During the reabsorption, the water and useful compounds will be reabsorbed from renal tubule to blood capillaries. But in the tubular secretion, the waste molecules, excess salts, these all from the blood capillaries again sent into the DCT region of the renal tubule. So that one we call as tubular secretion. And finally, the concentration of hypertonic urine. So once the tubular secretion over, the urine will be sent into the long tube-like structure we call as collecting duct. What we call collecting duct. The hypertonic, the formation of concentrated urine actually takes place in the collecting duct. I already told you at the PCT region only 75% of water will be reabsorbed, and at the loop of the nearly, because of the osmosis process, 10% of water will be reabsorbed into the blood capillaries. Still, what is the water present in the urine will be collected in the collecting duct. But here, the hormone, a special hormone known as vasopressin, it is secreted by pituitary gland. It is secreted by pituitary gland. That vasopressin hormone helpful to make the concentrated urine. So once there is a need to our body to secrete concentrated urine, it just released by our pituitary gland immediately, and the vasopressin converted the diluted urine into concentrated urine. So if you, for example, if you drink a lot of amount of water, there is no need of releasing of this hormone. Why? Because you are drinking a lot of liquid, at the same time you are excreting a lot of liquid. But really there is a need to secrete concentrated urine. At that time only vasopressin was released. What is the function of vasopressin? Means it just convert the diluted urine into concentrated urine. So if the vasopressin hormone is not released in sufficient quantity, it leads to a condition known as diabetes insipidus. Means they excrete the frequent amount of urination, means they excrete high quantity of urine. That condition we call as diabetes insipidus. Okay, now? And for example, next, then what is coming to human beings per day, they nearly excrete 1.6 to 1.8 liters of urine in normal per day. But it varies by and it changes according to the intake of fluids. If we take more fluids, the volume may be increased. If we take less liquids, the volume may be decreased. And the color of the urine is amber color. What we call amber color. That is because of the pigment known as urochrome. Urochrome is a pigment produced by the destruction of the hemoglobin in the liver. And the but slightly there is a denaturation of urea to ammonia, the conversion of acetic urine to alkali takes place. And in the persons, if there is a complete kidney failure, means if the kidneys are not working, we call that stage their body is filled with the waste molecules. That condition we call as ESRD, means end stage renal disease. End stage renal disease. And in the ESRD persons, their body is accumulated with the waste molecules and toxic materials. Such condition we call as uremia. Uremia patients become tired very fast and their body parts are swollen due to the accumulation of liquids. And for generally at normal, if the functioning of one kidney is not proper, the second kidney of your excretory system will be taken up the function of both. But even though one kidney is present, your excretory system is little bit okay. But if the two kidneys are not work properly, means the two kidneys stop their functioning, what happens? Your blood is not filtered, means your waste molecules in your blood and excess water, excess salts which are not removed from your body. So even it lead to the different conditions, it lead to the death also. So to avoid that, a special technique was developed by scientists that we called as dialysis. In the dialysis patient, the dialysis worked for the patients in those two kidneys are failed. So in those patients, the blood is taken from the main artery and mixed with an anticoagulant or anti-clotting substance known as heparin, and after that they send the blood to the dialysis machine. That dialysis machine actually takes up the function what actually done by kidney. So what is the function done by your kidney? The same function even done by your dialysis machine also. So once the blood is purified in the dialysis machine, the filtered blood will be again mixed with the heparin and anticoagulant and enter into your body to the vein. Okay, now? And even for example, dialysis is not a permanent solution. So dialysis until once the person want to alive. So throughout his lifetime he go to the dialysis procedure for every 5 to 6 days. So this is not permanent solution. So actually the dialysis we also call as hemodialysis. Why? Because here the blood is purified and filtered. That's why we call it as hemodialysis. But it is not a permanent solution. To improve that, the scientists try to transplant the whole organ actually, that we call as kidney transplantation. Kidney transplantation means just remove the kidney which was not functioned properly, and in that place the kidney which we get from another donor will be fixed. That process we call as, anyway you already know about organ transplantation, like heart was transplanted, liver, even in the same way kidney was also transplanted. So the kidney transplantation done by a scientist known as Charles Hufnagel in the year 1954. Kidney transplantation first done by a scientist known as Charles Hufnagel in the year 1954 between two identical twins. But coming to India, the first kidney transplantation occurs in the year 1971 at Christian Medical College, Vellore, Tamil Nadu. Coming to India, when the first kidney transplantation takes place means 1971. Not only the kidneys, still are in our body we have accessory excretory organs also, means additional excretory organs like lungs, skin, liver, even intestine also. For example, during respiration CO2 and water will be released, will be excreted by the lungs. And skin in the form of sweat, it excrete excess water and salts also. Coming to liver, the old RBC, means after the lifespan of 120 days, the RBC are destroyed in the liver. So during that process, some pigments like bilirubin, biliverdin, even urea was excreted through urine, and bilirubin and biliverdin are excreted in the form of bile along with the excess salts like K+, Na+, and even steroids, vitamins. These are excreted in the form of bile. And coming to intestine, from the undigested food, excess salts will be excreted in the form of feces. Then I already told you different organisms for the removal of the waste materials, they use different processes. Then compared to the protozoa, the excretory system is highly developed. For example, coming to protozoans, first we go for protozoans like amoeba and paramecia. By the simple diffusion method, they will be excrete the waste materials to the surrounding water from the body surface. Coming to next category, porifera and next phylum, porifera and coelenterata. Porifera and coelenterata, like sponges, these are. In these animals, the water baths almost also means they are completely present in water. So the water completely baths in their cells. So the waste materials will be sent. The next category we called as platyhelminthes are known as flatworms. So this one very important actually. So till the porifera and coelenterata, those are no specific excretory organs. But coming to platyhelminthes, first time the excretory organs known as flame cells was developed to send the waste materials. So in which class in the animal kingdom or in which phylum the excretory organs are first time developed is the platyhelminthes. What are the excretory organs? Flame cells. And the next one, nematoda. In the nematoda organism, the cells known as renative cells. Renative cells are excretory organs. Coming to annelids like earthworms, leeches, the nephridia are the excretory organs. The next one, like arthropoda, all insects contain either green glands or Malpighian tubules. From this topic, mostly they ask the weight. So in the arthropoda, what are the excretory organs? Green glands and Malpighian tubules. And after that, the mollusca, the metanephridia are the excretory organs. And coming to echinodermata. Echinodermata means mollusca means like snails, the mussels, these all. Coming to echinodermata means starfish, these all. So coming to let's see sea cucumber, starfish, these all echinodermata animals. In the echinodermata, water vascular system was present. And remaining all other animal classes like reptiles, apes means nothing but birds. Apes means birds, apes, mammals, including humans, we contain kidneys as their excretory organs. The next one coming to, so till now what we see everything about animals. So even the next is plants. Even the plants also produce waste, means obviously yes only. Why? Because for example during photosynthesis the plants produce oxygen as the waste product. It will be sent out to the stomata of the leaves and lenticels of the stem. But mainly plants they absorb more amount of water. The excess water will be thrown out from the plant body either by the transpiration process or by the guttation. Transpiration means evaporation of water from the stomata of leaves. Guttation means sometimes you see from like grass, leaves, small water droplets will be sent from the tip, that we call as guttation. So by the transpiration and guttation, excess water is eliminated from the plant body. And next one more important topic is the plant. Plants means, so plants are able to produce actually the metabolic activities compared to animals occur at a very slow rate in the plants. So the waste materials also produced in a slower rate, in a lesser rate in the plants than compared to animals. But in the plants, the waste materials mostly they are stored in different forms. In the different parts like fruits, leaves, bark, those all. So plants mainly able to produce two compounds by their metabolic activities. First one we call as primary metabolites. What we call primary. Plants are able to do types of compounds by their metabolic activities. So first one we call as primary metabolites like proteins, carbohydrates, fats, and these are used for general growth and repair of the plant body. But next coming to another category we call as secondary metabolites. The compounds which does not use it for general growth and repair of the plant body, we call them as secondary metabolites. So these secondary metabolites are of different categories. For example, first one we call as, actually these secondary metabolites plants produce for their own benefit, but even humans mostly we use the secondary metabolites in different ways. Means we are economically and in different ways we are benefited by the secondary metabolites of the plants. So first one coming to alkaloids. These are actually nitrogenous byproducts. These alkaloids has a wide number of uses. For example, here I don't have sufficient space to write. So for example, I tell some of you examples. For example, coming to alkaloids. First one, we use anti-malarial drug. Anti-malarial drug, quinine. We use quinine. Quinine is a alkaloid used as the anti-malarial drug. Quinine is taken from the plant Cinchona officinalis, and at the same time nicotine, one more alkaloid, it is present in the plant Nicotiana tabacum leaves. Even for example neem plant or Azadirachta indica. So that plant whole leaves, bark, stem, those all are used as in the secondary metabolite products. Okay, na? So those are the examples for alkaloids in the textbook, even the table. Just read the table, that is also very important. The next one coming to tannins. Tannins are the carbon compounds. So tannins mainly used in the tanning of leather, even in the medicines also. So the plants like acacia and acacia produces the tannins. The next one we call as resins. Resins are mainly secondary metabolites mainly present in the special passages of tubes known as resin passages of the gymnosperm plants. Gymnosperm plants means the plants which are having naked seeds without any fruit. So in the gymnosperm plants, in the resin passages, the secondary metabolites resins are present. These resins are mainly used in the preparation of varnish. Example, pines. And the next one, gums. So most of the time, especially in villages, on the bark of the trees like the neem tree, acacia tree, we see that a sticky substance oozing out from the bark of the tree. That sticky substance helpful to heal the wound of the plant. So whenever there is any branch cut, the gum was produced. That we use it as economically as adhesive and binding, used even in the preparation of foods, even in the preparation of medicine, those are. And the next one we call as latex. Latex is a milky white substance. Latex is a milky white substance that was produced by different plants. For example, Hevea brasiliensis. Even in normally we call it as rubber tree also. The Hevea brasiliensis produce the latex. From the latex of the Hevea brasiliensis or rubber plant, we prepare the rubber. And the next one, the latex of the Jatropha plant, we use the latex of the Jatropha plant we used as the biodiesel. And the next one, Bregman, a scientist who tells that the plants not only absorb the fluid from the soil, that at the same time they release some of the peculiar secretions or some of the waste secretions into the soil where they are growing. So even we see the same thing. For example, if we grow the either apple crop or any crop for several years in the same soil, after some years the soil may not suitable for the cultivation of any fruit plants or any normal plants. Why? Because it released some secretions into the soil. Because of that, the crop yield was decreased. So basing on that, we say that the plants secrete some waste molecules into the soil, and some of the waste materials they store in different forms like fruits, leaves. Whenever the leaves and the fruits ripen and the leaves are become older, what happens? These will be separated from the leaf separated from the plant. So even the waste molecules also get rid of from the plant body. So thank you students.